Our first speaker today is Ashwin Narain. Ashwin started his career in science with a bachelor in biotechnology at the National Institute of Technology in Warangal in India. After receiving his degree, he moved to the University of Würzburg in Germany for his master's, where he is now doing his PhD in biochemistry and molecular biology in the lab of Elmar Wolf. He's focusing on transcription and in particular on the elongation factors SP5 and SP6, which I guess we'll get more about in a moment. So Ashwin, we're really happy to have you here, and the floor is yours.
Okay, I hope I'm loud and clear. Yeah,
we can hear you perfectly.
Cool. Thanks a lot, Matthew, for the nice introduction. And so let me get started. Okay, cool. So thanks a lot again for the Fragile Nucleosome community and the organizers for this really nice opportunity for me. And I'm really thankful for the Discord server, and it has been a really nice journey so far to interact with this vibrant scientific community. So I'll be talking about a recently published work from our lab on SPD6 and its role in RNA-POL2 elongation. As introduced, I'm working in Elmar Wolf's group, and we are housed in a small but a beautiful city of Würzburg in Germany. As a lab, we work on the interface of MYC, transcription, and cancer. And when I joined Elmar's group back in 2018 as a master student, a couple of interesting findings about MYC, the oncoprotein, were being explored. A couple of grad students observed that MYC affects RNA-POL2 transcription by associating with elongation factors like SPD5 and SPD6. Now, whereas the story on SPD5 got published back in 2019, I got interested in SPD6. Now, what is SPD6? So SPD6 is a histone chaperone and lactate complex. It is involved in the reassembly of nucleosome post-transcript production. And in the absence of histone chaperones, the transcribing RNA polymerase II could run into the nucleosome, leading to chromatin disintegration and eventual transcriptional chaos, which indeed is the case for SPD6, as reported back in 2003 by Fred Winston and also in 2018, that in geese, that in SPD6 wild-type strains, transcription initiation happens from the canonical transcription start site with the binding of general transcription factors. However, in loss of function SPD6 mutant strains, the general transcription factors bind intragenically, and that is primarily leading to the cryptic intragenic initiation. This intragenic initiation is actually happening because of the loss of chromatin organization in the absence of SPD6. One more thing further got us interested into SPD6 was that SPD6 also stimulates RNA-POL2 elongation in non-chromatinized templates. Furthermore, and more importantly, recently, as it was reported by Cecil Foss when she was at the Patrick Kramer's lab, that SPD6 tightly interacts with the elongating form of RNA polymerase II. Now, this suggests that SPD6 probably also has some direct effects on transcription, which are independent of its role as an epigenetic safeguard. So we asked a couple of questions. First of all, what is the role of SPD6 in RNA-POL2 elongation? Second, does SPD6 suppress intragenic initiation in mammals also, as observed in yeast? And third, are there other direct roles of SPD6 in transcription which are unknown still? As we wanted to aim for the direct effects, we thought of acutely depleting SPD6 using the auxin-inducible de Graunt system. To do that, we first of all tagged SPD6 at the C terminal in U2OS cells with the AID tag and a V5 tag, and then a self-cleaving peptide with the blastocidin as the selection marker for the knock-in. The full membrane blots over here reveal that this shifted running pattern, which one would expect for a tagged protein of interest. Strikingly, within 4 hours of auxin treatment, we could clearly see the levels of SPD6 completely abolished. And that was also replicated in another independent clone, which we also used for our study. We also wanted to see the binding of SPD6 on chromatin. For that, we carried out ChIP-Rx, the spike normalized ChIP-seq. So what you see over here in control condition, the blue, which will be always the control condition in my talk, and orange will be the auxin condition. You can clearly see that SPD6 starts binding after the RNA-POL2 pause release. and it's evenly binding throughout the gene body, then accumulating after the end site, similar to the RNA pol2 elongating behavior. As expected, after auxin treatment for 6 hours, we clearly see the the sp86 binding on chromatin is completely abolished. And once we had the system, we thought of asking the first question, which was that what is the immediate nascent RNA transcription changes on after sp86 depletion? For that, we carried out Fourier sequencing, in which we first treated the cells for 6 hours with auxin, and in the last 15 minutes, we labeled them with Fourier. Following Fourier pull down and sequencing, we got the results over here, which you see in the form of a metagene. In control condition, we could clearly see for all replicates, for the triplicates, we clearly see in case of the reads are evenly distributed throughout the gene body, as one would expect, and then they sharply drop at the end site. However, in case of auxin depletion, that means in case of sp86 depleted condition, we could clearly see that in all the replicates, the reads are sharply dropping as we moved into the gene body, pointing out that there seems to be an elongation defect. We wanted to know it further, so we looked at individual genes. Over here, you see an example of a very long gene and a medium gene. Whereas the unperturbed cells have even distribution of the reads, in case of sp86 depleted condition, what we observed was that the reads are only appearing like in a fraction of gene, which looks like that RNA pol2 is probably productive only until a fraction of gene, and then going back to the background levels. Interestingly, this similar effect was also observed in medium length gene. However, in this case, the reads are still appearing towards the end of the gene. So it seems like there is an effect which is accumulating over the gene length. Now we wanted to quantify it and further analyze it. For that, we collaborated with mathematical modelers. The mathematical modeling suggested that there could be 2 defects which could lead into such an observation. First would be the processivity defect, and the another was elongation rate defect. Now, to distinguish between these 2 possibilities, we did a couple of more important experiments. First, to observe the occupancy of RNA pol2 with the total and the phosphoserine RNA pol2 chip-seq. We also observe elongation rate using DRB for ASO-seq, the transcription rate using SLAM-seq, and eventually did multi-omics to observe what are the transcriptional dynamics. So I'll share data of the chip-seq, for which we treated the cells again for 6 hours with auxin, and we did an IP against the phosphoserine 2 CTD pol2. In case of control condition, we could clearly see as an expected pattern of phosphoserine to the elongating form of pol2, that it evenly binds in the gene body, and then it sharply increases the pause, denoting this 3' pausing, which is a feature of the RNA pol2 transcription. However, when sp86 is not present, you clearly see a very drastic drop in the reads of pol2. That means the occupancy of pol2 is dramatically reduced as we move into the gene body. This points out that it is the occupancy of pol2 that is getting lost as we move into the gene body, which means the processivity of pol2 is probably getting lost. That's why it's getting stuck and falling off the chromatin, giving us a conclusive insight into the mechanism behind this elongation defect. So to summarize the first part, we indeed observe that acute loss of sp86 interferes with transcription elongation. And our data suggests that this results from a major processivity loss and a mild elongation rate defect, the data which I have not shown due to time reasons. I would also like to point out that similar results were also published by Patrick Kramer's lab in the same issue of Mol Cell, but using a different acute depletion system on sp86. Moving further, if you look back at our Fourier data, we couldn't really observe any such intragenic initiation kind of phenotype or any sort of accumulation of the reads. We wondered why could that be the case, because the protein levels are completely gone. We thought that could it be the sustained depletion of sp86 that might be the reason. For that, we thought of using silencing sp86 expression using an SI against it for 48 hours. Why we did not use auxin was that because after 24 hours of loss of sp86, cells were starting to die. So using the system, we again carried out Fourier sequencing And forensic analysis revealed that there is a massive redistribution of reads in the sustained loss of SPD6. We further characterize these peaky regions over here using motif analysis and also general transcription factor ChIP-seq, and that revealed some signs of them being initiation sites, similar to what has been observed for yeast. To see whether this phenomena happens globally or not, we called these peaks, and you could identify about 21,000 such peaks in case of sustained loss of SPD6. Remarkably, though, when we tried finding this, we couldn't find any such initiation sites in case of oxygen-depleted condition. Bringing us to the conclusion of the second part, we think that in contrast to sustained depletion, acute depletion does not cause intragenic initiation in mammalian cells. Which brings me to the model that we have for the first part of the talk. We think that our data suggests that SPT6 maintains processive elongation and epigenetic integrity by allowing the passage of RNA pol2 across the nucleosome. By that, it also maintains the nucleosome integrity, and thus the epigenetic integrity is safe. In sustained loss of SPD6, however, the transcribing pol2 runs into the chromatin, and that leads runs the nucleosome, and that leads to the disintegration of the nucleosome. This loss of nucleosome leads to a naked chromatin, and that would eventually lead to epigenetic chaos, which is observed in the form of intragenic initiation, similar to yeast. However, in pretty much contrast to sustained depletion in acute loss, RNA pol2 is not able to make through this nucleosome barrier itself. Now, there are two, and that leads to unprocessive elongation. And there are two explanations which could be there for that. First of all, as SPD6 can bind to histones, it can aid the passage of pol2 across the nucleosome, as also proposed by Patrick Kramer's lab. However, there's also another possibility that there could be certain unknown allosteric functions or recruitment functions, because SPD6 also promotes elongation in non-chromatinized DNA, a theory which we are still following up, which might also affect the processivity. Coming to the last part of my talk, we were also wondering that are there any direct effects on transcription which are still unknown? There indeed were some of them. So if you look back at our forensic data, what you see over here in example gene is that in unperturbed cells, the reads are distributed evenly throughout the gene body, whereas as you move outside in the transcription end side, they drop pretty sharply. However, if you look at the oxygen condition, SPD6 depleted cells, the reads are still coming up even several kbs downstream of the end side. And that is a phenomenon of read-through transcription. This further happens globally if we see over here in this heat map for all triplicates, in which red denotes more reads in oxygen and blue denotes more reads in control. More importantly, we observe that longer genes seem to be not affected by this. And the reason why we think that is happening is because there's a drastic processivity loss in pol2, and none of the pol2 is actually able to make to the end of the gene itself. Whereas in case of the shorter genes, a lot of pol2 molecules is reaching to the end, but it's not able to probably terminate. Now, because lack of termination is there, we thought that 3' pausing is an important feature of RNA pol2 termination. So we thought of looking at that using our data set of phosphoserine to pol2 ChIP-seq. In the unperturbed cell, we clearly see this 3' pausing happening around the end site, which is a feature of RNA pol2 transcription. However, in the oxygen condition, we clearly see that there is no such pausing happening. And more importantly, after this pausing region, we also see a lot more pol2 molecules accumulating if we compare to the control condition, explaining that these pol2 molecules are still elongating, and they result in this read-through transcripts that we observed earlier. And this phenomena is globally happening. Now, because the termination machinery seems to have gotten affected, we thought that RNA pol2 interactome might have also gotten affected. To see that, what we did was, we looked at the RNA pol2 interactome in the presence and absence of SPD6. And for that, we overexpressed an HA-tagged RPB3 subunit of the pol2 complex. Then we isolated only the chromatin fraction and did an HAIP followed by mass spec. What you see over here in the volcano plot is that SPD6 is the strongest dropout. As it is depleted, we expect that. More importantly, XRN2 and symplecin, other transcription termination, canonical transcription termination factors are also dropout, showing that termination defect is indeed real. Surprisingly though, the CPSF complex, cleavage and polyadenation complex, is not changing much, whereas the CSTF complex looks like it has changed much, all three subunits. Whereas we are following up on the idea that CSTF complex might be responsible for this effect. In literature, there seems to be some hint towards that back in 2015, as published by Proudfoot Lab. They also observed similar defect of read-through transcripts when they silenced CSTF64, which is a synonym of CSTF2. Which brings me to the last part of to the conclusion of the last part. We think that SPD6 is essential for RNA-pol2 termination globally. And that probably it does by supporting association of specific set of termination factors around the polyase site. So to summarize my study, I would like to summarize in the form of an mRNA genes race course. We think that the horse over here, the pol2, requires SPD6, the jockey, for completing the race efficiently and also stopping so that termination defects don't happen. But there's a twist there. We have a lot of hurdles here, which are the nucleosomes. And when I come into the picture and acutely remove SPD6, RNA-pol2 runs into the nucleosome and loses its processivity. Furthermore, if this happens for multiple laps, that leads to disruption of all these hurdles and eventually a chaos, the transcriptional chaos. With that, I would like to thank everyone involved in this study, from the Cancer System Bio Group, Elma for being a brilliant PI, Pranjali and Apurva for helping with the nascent RNA experiments, bioinformatic analysis and visualization and the help during the project, Bikash for helping with the cell line generation, and all the other members of the Arge Wolf group for a lot of support. Florian for helping with the mathematical modeling part, and the other collaborators in the University of Würzburg for different aspects of the project. And thank you all for your attention, and I'm happy to take any questions.
Thank you, Ashwin. Very nice and very clear talk. It was great. So we already have some questions coming in. Just a reminder for those that joined late. If you want to ask questions, please ask them in the Q&A box or raise your hand so we can unmute you and you can ask the question by voice, which is what I'm going to ask Arvind to do. So you should be able to unmute yourself now.
Hi, Ashwin. It's a nice talk. Can you hear me?
Yeah, I can hear you. I can hear you clearly.
Great. So sorry if I did not get the first part, but what I got, you mentioned that SPT6 loss cause loss of nucleosome, right?
Yeah. No, it doesn't cause loss of nucleosome. The point is that maybe you can complete the question, then maybe I can explain.
Okay. Because my question is in terms of the mechanism. If SPT6 mutation in yeast that you showed, if it caused the loss of nucleosome, then how it affects the RNA polymerase elongation? I found it's like counterintuitive, because if there's a no nucleosome, then the elongation is supposed to be like more smooth, right? So why SPT6 loss cause defect in elongation if SPT6 loss cause loss in nucleosome?
So first of all, so as I said, we could differentiate that in acute loss of SPD6, there is no such loss of nucleosome happening, okay? And this is why we think that it's a sustained loss of SPD6. And that's why what I tried to show, that only in sustained loss of SPD6, the nucleosome will be lost. If we observe the effects within 6 hours, we do not think that a nucleosome is lost right away. We think the processivity is getting affected by something else. And that probably is, as also shown by Patrick Kramer's lab, is that this ability of pol2 to go across the nucleosome. Because in the naked DNA, it's not able to go through the nucleosome, and that's why its processivity getting affected, and that's why the elongation is getting lost. So nucleosome loss happens really later. For yeast, I cannot comment completely on that because, that I cannot really comment completely.
Okay, got it. My second question is about this. Looks like it is affecting RNA polymerase elongation. So why only Pol2? Why not the other polymerase elongation? Why it is not affecting other polymerase elongation, including DNA polymerase as well?
That's a good question, but we have not looked into it. It could have affected, but I cannot comment because we have not looked into any of the Pol1, Pol3, or DNA Pol. I cannot comment on that. It could well be.
Okay. So this mutation, is it non-viable?
Arvind, I'm sorry. Would you mind joining the coffee chat afterwards for the questions? Thanks a lot. Because we have a few other questions. One is from Michael Levin, who's asking, could we comment on the status of Pol2 in chromatin after the oxygen-induced sp6 degradation? We're getting probably at a similar point. Do you have Western blot for the level of Pol2 ubiquitination?
Okay, yeah, I think I get the question. Yeah, the status of Pol2 in the chromatin. With the ChIP-seq, we could observe the redistribution. Yes, the levels of Pol2 were not affected much using Western blot. Whether ubiquitination of Pol2 is getting affected, we did not check it. We did not check that. That I can say that. But yeah, the levels were not affected for the next because the time period which we used, no, the levels of Pol2 were not affected in our case.
Okay, then we have one from Corin Logie. Could it be that the read-through transcripts are in fact spurious RNA Pol2 initiation at the 3' end of the gene? So basically you have more initiation at the end of the gene, and that is why you see the XR reads after the 3' end.
I mean, I would say that, as I said, we are observing the effects after 5 to 6 hours, right? So I'm not really certain that could be initiation within this short time period. So I don't really think so. I think it definitely is read through because of this moving Pol2. I don't think so that it would be the spurious initiation.
Okay.
Wrong, but I think it's not.
Yeah. Then I'll ask the last one from Benjamin Sabari. This is nice talk. Do you find any change in the NLEF or PAF1 subunits in the interaction with Pol2?
Yeah, actually we do find subunits of NELF. Which one exactly I do not remember, but yes, we do find subunits of NELF. They did lose interaction with Pol2. Yes, they do. PAF I cannot comment because I think we couldn't find a couple of components due to some mass spec issues. I would say that because we cannot find all of them. But yeah, for NELF I can say that yes, some of them did lose interaction. You can find it on our paper. Yeah, the details are there.
Great, thanks. So because of time, I think we have to stop here with the questions. But again, please, if you have further questions you want to go with Ashwin afterwards, please let us know in the chat box. And at the end of the session, we can after finally enter into this discussion.